A reminder that in this channel, we mainly talk about neuroscience, more specifically in the electrical activities of neuroscience, aka EEG, brainwaves frequencies, and in addition to that, we talk about future of AI systems as well as humanoid slash robotics. And so sometimes the research papers that I share with you here can be a little bit technical and most probably boring if you're not interested in this topic. So I just want to put it out there because some of you don't seem to maybe like some of the topics, but I'm just letting you know that this is the whole gist and the, I guess, goal of this channel. And in addition to that, we talk about symbiotic intelligence, where it's a combination of how the brain works in its analog way, trying to mash that up with the digital systems such as humanoids, future of AI, as in AGI and ASI, most of which are still speculative, but the nature of science is that we are speculator first and foremost, at least in the beginning, and then together we do research and studies and stuff like that. I also want to mention that sometimes I do introduce some tools if I have found them to be helpful. For example, most recently, the Thinking Machine startup announced Tinker, and it is not quite launched yet. It's still in beta, and I don't even have access myself, but I'm sort of poking around with it. Also, sometimes we talk about Python, and I think Python is great. If you're a neuroscientist, it's great that we all start learning that a little bit. My background on it is extremely at a sort of a beginner level, but that's also another thing that I think we should all think about in terms of sort of improving our skills, especially if you're a neuroscientist or an aspired one. So with that, let's go ahead and start. There was a fascinating new study that just published as preprint. And as always, I share the link below for your reference, that they talked about looking at the neural oscillation, aka brainwaves, in a different manner. Mostly when we look at EEGs or LFPs, we look at sort of a wave of brain electrical activities at a sort of eagle eye view, if you will, or bird eye view to some extent when it comes to LFP. But in this case, they went more into zooming in into specific parts of the brain and looked at the brainwaves frequencies that way instead. And as a result, they share their findings with us here. And they also talk about these rhythmic patterns of electrical activities of the brain to be sort of like packages, and they also make some connection with the action potential switch on and off of the neurons itself, and how that then in turn also exhibits electrical activities. At the end, we will also look at these results and, as always, try to connect that to potentially the notion of symbiotic intelligence, where we connect neuroscience with our future of AI. So this preprint introduces a novel way of studying brain rhythms, aka brain frequencies, by focusing not on field potentials, which are the traditional approach as in action potentials, but on the spiking activities of neuron itself. Neural oscillation, or brainwaves frequencies, are the repeating rhythm patterns of the brain activities, the electrical activities, and they have long been known as a central way to look at how brain coordinates information and communication within different regions as well as different timescales. Most research has captured them through, for example, EEG or LFPs, which measure the collective electrical activities at the tissue level. However, Oscillations, aka frequencies, are also present in the timing of the neuronal spikes and how they propagate across brain regions and how they connect to behavioral as well as physiological measures. The authors of this paper propose that by looking directly at the pooled spiking activities of large number of neurons in a local circuit, one can uncover these rhythms, and importantly, we can isolate individual cycles within these neuronal activities. And so traditionally, when we study brain waves or electrical activities of the brain, we usually use methods such as EEG or LFP. But the problem there is that when we do so, we're looking essentially at activities about thousands or maybe millions of neurons. We're never able to go into individual brain activities of individual neurons. And that's why feels more like you go into a stadium and look at and you hear everybody's cheering, but you don't know how the individuals are reacting within that stadium, for example. In this particular paper, these authors point out that there is another way to study these rhythms by looking at the actual spikes fired by the neurons itself. A spike is actually the tiny electrical signals that a neuron sends when it is active. But these spikes also carry rhythmic patterns. It's not just that they spike because it's an action potential. And until now, there haven't been a clear way to tell whether these rhythms in spiking match up with the ones that we usually measure in EEG and LFP. Remember, EEG and LFP is when we look at brainwaves activities at a larger scale level. There are thousands and millions of neurons involved, but we were never able to sort of connect them. Are they connected to the action potential that happens when a neuron is on or off? And that is why this particular paper is so exciting, especially for a person like me, because the voice of the individual neuron itself might tell the same story as the crowd noise of millions and thousands of neurons. But do these rhythms spread across different brain regions in the same way? That's the question. And how do they link to what, for example, a person or an animal is doing or feeling in real time? The authors argue that by pooling the activity of many neurons in a local brain circuit, that is looking at the spikes of a group of cells together, we can actually uncover clear rhythms, clear frequencies, and not just general rhythms, but the individual cycles that make them up, like isolating each individual wave in the ocean instead of, for example, measuring the tide itself. The ability to zoom in on a single cycle opens up a whole new possibility, because it means that scientists now can study how specific brain frequencies rise, fall, and interact with each other on a moment-to-moment basis, giving us a much sharper picture of how the brain organizes its activity across time and across brain regions as well. The second question then focused on how oscillatory spikes, the neural oscillation, correlate across brain regions at different time scales. By comparing cycles isolated from different regions recorded simultaneously, the scientists showed that a delay in population spiking between brain regions are not fixed, but instead depend both on which regions are communicating and on the frequency of the rhythm itself. Faster rhythms, for example, propagated differently across brain regions than the slower frequencies, suggesting a time scale specific structure of inter-regional communication within the brain. This adds an important dimension to how we think about large-scale brain coordination, in that it reveals that the temporal organization of communication is more dynamic and layered than we previously anticipated. One of the findings of this paper was that surprisingly, the timing of these delays was not fixed. It depended both on which brain region were the ones that were talking to each other and on the tempo or speed of that rhythm, of that frequency. For example, faster rhythms like gamma waves, which are the highest frequencies, spread to the brain differently than slower rhythms, let's say alpha brainwaves frequency. And they could last for seconds or even minutes, these faster frequencies. In other words, the brain does not appear to communicate in one size fits all way. Instead, each time scale has its own rules for how signals are traveled and aligned between different brain regions. This is important because it shows that the brain communication is more flexible and more layered than we used to think. Instead of imagining the brain as a one big orchestra where all the instruments follow the same conductor, it's more like multiple ensemble playing at different speeds, each with their own timing and their own coordination rules. This insight changes how we think about large scale brain activity, because it means that the brain's network organizes itself and themselves differently depending on the frequency they are using to share information among themselves. The authors of this paper essentially designed a machine learning approach using, for example, some specific model, and they pre-trained them on synthetic data, which can reliably detect and align single cycles of frequencies in spiking activities. Then they validated this method in recordings, for example, from mice, showing that it successfully identified frequencies ranging from fast gamma waves all the way to ultra slow waves lasting hundreds of seconds. But the ultra slow rhythms here were not only visible in spiking, but also tracked in behavioral measures like pupil dilation and facial movements in the subjects, which are well known indicators of, for example, the various emotional states within subjects. These findings suggest that population spiking of neuron activities contain a richer picture of brain frequencies than field potential alone. Field potential is when the neuron spikes, it goes basically off and on, especially at lower time scales where field potentials tend to underrepresent activities. And the authors then applied this technique to address two specific scientific questions. The first one was that they examined how to better align neural responses across repeated trials of visual stimulation. In this particular study, sensory input takes slightly different amounts of time to travel from the retina to the visual cortex across trials, which then created biological timing variability that can blur the results when responses are averaged in the usual way that we always do in scientific papers. But by isolating gamma cycles in spiking activities, they were able to realign trials more accurately, reducing the noise caused by these transmission delays and providing a clearer view of cortical dynamics during the sensory processing. This analysis supported the view that sensory processing is organized into discrete rhythm packets rather than being continuous. a finding that deep. Okay, now lastly, let's see how slash if this could be another finding that could potentially be used and addressed in the symbiotic intelligence work. And in that, to some extent, it resonates because with symbiotic intelligence, we talk about incorporating real time data, as in the electrical activities of the brain, incorporating that into the future of AI systems. And so, whether it's a biological or artificial intelligence, these findings emerge from the dynamic coordination of many interacting units rather than from isolated computations. For example, what these authors show is that the brain's communication is not rigid, but it's a time scale dependent with rhythms or frequencies of different speeds, shaping how brain regions align and exchange information. This layered, flexible structure could potentially serve as a blueprint for the future of AI systems instead of designing systems that process information in purely linear or uniform fashion. We could create architectures potentially where different modules communicate at multiple temporal rhythms, each tuned to specific functions, specific coordinators, and specific rules, so to speak. Fast cycles of rhythms might handle sensory input or real time responses, while low frequencies could potentially govern long term planning, memory integration, or shifts in internal states of the subjects. In this sense, the brain demonstrates that true intelligence emerges not from raw processing power alone, but from the symbiotic interplay of rhythms, frequencies across scales. Applying these principles to future of artificial systems potentially could bring us closer to machines that don't just compute but also think, adapt in a way that mirrors the biological human intelligence. Okay, guys, that's all I had for today. I'll link the paper below as always. I should also correct myself that this is not a preprint, this is actually published in computational biology. But the link is below if you want to read it for yourself. And it's a very interesting, they have a lot of details there that I'm sure you find useful. But again, if you are a neuroscientist, that's probably interesting to you. But if you're a computational, if you're an engineer, software engineer in LLMs or other AI topics, this is probably a little bit too details for you in terms of neuroscience, but I try to combine it so that it would be enough summary to give you ideas if you don't have neuroscience background. Okay, thanks again, and I'll see you guys next time. Bye.